So first of all, thank you for giving me the opportunity to talk about RNAscope today. I guess Jake Estes was not available, so you stuck with me. A few years ago, Jake and I adapted this really interesting method that ACD Bioscience developed, and we're called the next generation of in situ hybridization. And I just want to walk you through really quick the different big steps of this new in situ method. So first of all, you have to access to your target. So for that, either you're on cells or FFPE tissues or OCT tissues, you have to access your RNA or your DNA. For that, we're either heating and or treated with protease. And then we put the probe on. We hybridize the probe, and this probe is really specific. That's what makes the RNAscope so specific, is the fact that you have to get your two Z pairs right next to each other to actually start your amplification steps. There is multiple steps of amplification that makes the technique extremely sensitive, and we're capable to actually detect down to one double Z pair in the tissue. Then you visualize and you quantify, and I will also spend more time on the quantification. I think it's really important, and there is a lot of improvement to be done on that. So here's how it looks like. I'm pretty sure you've seen similar pictures before. So this is an example of a rhesus monkey lymph node that is highly infected. The first picture is RNAscope. If you look closely, then you will see that there is different patterns. You could detect productively infected cells, and also we are in the B cell follicle, and all of those little dots are actually variants that are trapped into the FDCs. The second picture is difficult to see, but it is DNAscope. The signal is way smaller, but all those tiny little dots are a signal for viral DNA. And we could combine the both. We could combine RNAscope and DNAscope, and then visualize the infected cells, the virion, and the cells that are also harboring viral DNA. So this assay is really changed the way of doing in situ. It is a single day assay. It's highly sensitive and highly specific. And we could target different clades, different viruses, including chimeric viruses, or exclude different part of the genome. And this is an example on how sensitive this technique is. Here on the upper panel, you have representative pictures in a high viral load monkey. And we could also detect viral RNA and obviously viral DNA in actually fully suppressed animal or patients. What people are more and more interested about is actually to phenotype and characterize more the cells that are harboring viral RNA and viral DNA. And for that, there is an easy way to do this. We are combining, we're using fluorescence approach, the RNAscope. You have here the viral RNA in red, the CD4 T cells in green, and the myeloid lineage characterized by CD163 and CD68 in blue. And the picture right here is an example of viral DNA expressed in an IL-10 positive cell. Sorry. So just to give you a few examples on how we're using RNAscope approach into the EDGE research. And this is a paper that we published recently with Brendan. So here our goal was to understand the HIV transmission, and more specifically the really first events that were happening at the site of infection. And for that, we used the NHP model, and we used one of the great tools that Brendan developed, the HIV MAC239X, so a tag virus. And we scheduled necropsies at different time points post-infection from 3 days to 14 days. And we wanted to go as deeply as possible into the tissue to try to characterize those really first events of infection. And for that, we decided to combine laser capture microdissection and RNAscope. So we took different sections, up to 10 sections of 7 micron each. 2 of them were used to actually kind of develop a map of where the virus were, and we saw really clearly those lymphoid aggregates at the site of infection. So we identify each little area, each little lymphoid aggregates that were positive for RNAscope, and capture them using the UV laser cut. So each area were on one cap. Then we extract the DNA, and Brandon's team performed the SGA and the MySeq analysis. So what we found was pretty interesting, even if it probably kind of makes sense for a lot of people, but we were able to show that there was same variant in multiple lymphoid aggregates, so which means that all those foci are actually linked to each other. And we also saw that variants setting local infection. So every variant that were found within the female genital tract were actually also found in the blood. Another way that we used DNA, this time DNAscope, was for HIV reservoir, and we collaborate with Mirko on this project. And here's some example of the pictures that we generated in rhesus monkey that were treated. We stained for viral DNA, CTLA4, and PD1 within the T cell area and cell follicles, quantified the cells that were harboring viral DNA and that were either CTLA4 positive or PD1 positive or negative. And what we found out is a set of those CD4 memory T cells that are expressing CTLA4 and are not expressing PD1 actually crucial for viral persistence in treated monkey. One last example for the application of RNAscope in the AIDS research. This time, this is a collaboration that we have with Sharon Lewin in Melbourne. And this time, we did not target the virus. We actually helped her to understand the correlation with CXCL10 and the fibrosis in her cohort. So the goal of this study was to determine whether the liver disease was associated with intrahepatic or circulating markers of inflammation, or it was due to HIV or HPV burden. So in this cohort, there was 39 participants. All were co-infected with HPV and HIV. And for now, this paper that we recently submitted to PLOS Pathogen was only with patients prior to ART initiation. And they performed liver biopsies and collected blood samples. And so what Sharon and her team showed was that the liver fibrosis was statistically significant. There was an association with the intrahepatic mRNA and for CXCL10 and CXCL3 and the HIV DNA. So what was the role and what we did on this project using the RNAscope approach? We actually stained for CXCL10, quantify the expression of CXCL10 within the liver biopsies in the co-infected patient versus we had a few negative liver biopsies. And we showed clearly highly expression of the CXCL10 within those liver biopsies. And the expression was actually really locally detected and was within the infiltrate right next to the portal tract or blood vessels. We also were able, by combining RNAscope and IFA, to identify the type of cells that were expressing CXCL10. And even if we had a handful of myeloid cells and neutrophils that were expressing CXCL10, the major population that was expressing CXCL10 was the hepatocytes. So RNAscope and DNAscope is not only for targeting HIV or SIV. Basically, if you have a sequence, we could target it. Those are just examples of what we done in Zika in the brain. We did on Sharon's patient also DNAscope on HBV in human liver, and we are developing a broad different probes for gamma herpesviruses for human and monkey samples. What I want to spend more time on, though, is the image analysis. And I think there is a lot of improvement to doing that. So in my opinion, a lot of people could actually generate really nice pictures like this. But how do you switch from a representative picture to actually numbers and a graph? So we spent years trying to dig into the many software that are out there. There is tons of it, and there is more every single day. But we decided to go with CellProfiler. That was one of the best software to answer our question and actually our needs. And so here's just quickly how we are doing this. We have a full section. We are picking region of interest, so ROIs, either in the T cell area or by drawing the entire follicle. We're extracting those pictures. So this is an example of a region of interest that we will have. And then, so by either using a predefined algorithm in CellProfiler or because this is a really flexible tool, you could set up your threshold and write your own algorithm. We are separating here, this is in red, so the rapid staining, which will consider and being detected a positive signal, obtain an outline, and quantify this. The good thing about CellProfiler also, and we've tried many softwares out there, this is the most accurate method that we find to actually count the nuclei, and so that those both algorithms combined allowed us to give you results for RNAscope and DNAscope by number of productive cells per nuclei or area, and also the number of virion per nuclei or area. The thing that we noticed really quickly, though, there was some limitation to CellProfiler. It's not only CellProfiler, there is limitation to a lot of software that we use so far. This is an example of trapped virion within the FDCs, and I hope you could see clearly that it's actually not a productive cell. The productive cells are way darker and way more intense. Those are virions. However, CellProfiler was actually recognizing this aggregate as a productive cell and was counted as a productive cell. There was another limitation is when the virions are really small and not maybe perfectly in focus, then CellProfiler was not detecting those virion either. So we decided to go with the expert, and we contacted Dr. Yanling Liu, that is working at the Advanced Biomedical Computational Science at the Frederick National Laboratory. And what they're doing is they are the expert in deep learning. So we spend a lot of time trying to train basically a machine. I'm not going to go through all how deep learning is working. I'm going to be really bad at explaining it anyway. So, but basically, you're feeding the machine with hundreds and thousands of pictures, and you QC this recognition of signals after. And so what we came up with is what we call the web app. I know this is a terrible name. We really have to come up with something else. If you have any idea, I take it. But the web app could be really used by a 4-year-old. This is extremely simple, and we really hope that we will be able to have more, as many people as possible using it, so we could actually compare our results. So here, this is an example of a B cell follicle region that we extracted. You have, sorry, it's difficult to see, but the productive cells here are actually even squared in green, and everything that is considered as virion is squared in orange. And you could have the exact position of every signal. You still have some control into that. You could change the size of the pixels, and you could change the roundness of the cells. This is another example of a much more busy slides, I will say, a much more busy B-cell follicle. And again, it's doing a fantastic job at recognizing the cells and the trap variant. So we needed a project to actually validate this new web app. And we had the perfect project for that in the lab. And I still don't really know why we started that, but we started it, so now we have to finish it. We call that the Full Lymph Node Project. And I think it's more than time to do that now with the tools and the approaches that we have. So I think we all know that lymphoid tissue are the main site of replication of HIV and SIV. However, right now, I don't know if you know, but most of the time when you send your tissue, your fixed tissue to a lab to do any type of analysis, the results are going to come back to you are performed on a 5-micron section, basically. So we wanted to know if 5 micron were actually representative of a full lymph node, if there was variation between the follicles and between the T-cell area, but also if there was variation across the different sites. So is a mesenteric lymph node the same thing as an axillary lymph node? And I think it's kind of established that pretty much everything is the same, but we haven't really looked at it. So we started with animals that were highly infected. We are also in the process to do that on treated animals. But we came up with a full panel of different markers. I will only talk briefly because those are really preliminary data of what we obtained with our RNAscope and target cells. And this is just to show you multiple sections across the lymph node. We're up to 600 or 700 sections per lymph node. And I think you don't even need a tool to actually already see that there is variations between the sections. We were able to compare our results obtained by CellProfiler compared to what we were obtaining with our web app. And we're clearly better at recognizing variants. And for the productive cells, this is the same thing here. We have a nicely better recognition of the cells in the T-cell area. And this difference that you see in the B-cell follicle is simply because CellProfiler was actually mistaken some of the trap variant as productive cells. So we have a much more accurate quantification way using the web app. In those animals that were highly infected, we saw that there was a number of viral RNA positive cells that was higher in the T-cell area than the B-cell follicles. And again, those results will be done in different monkeys at different types of the disease. Just a quick view at comparing different sites. Here you have the variant quantification by the web app, axillary, inguinal, and mesenteric lymph node within the B-cell follicle and the T-cell area. And you could tell that there is variations between those different sites, especially in the T-cell area. Actually, this is the same pattern, which makes sense. We had a perfect correlation between the number of variant and the number of viral RNA positive cells. But you could tell clearly that there is variations between those lymph nodes. This is just the CD4 T cell and myeloid cell data. There is much more to come. I don't want to go too much in detail, but there is also variation depending on where you are. Each dot is the average of all the ROI that we're obtaining from one section. So in conclusion, because I'm late, sorry. So I think I don't have to try to convince anybody. I think RNAscope is an extremely specific and sensitive approach that could provide really important information for the location, but also the phenotype of the cells that are harboring viral RNA and DNA. And this is a powerful tool to study HIV and SIV reservoir. I think using accurate quantification tool is essential to compare and interpret in situ data. And I really hope that more people will use the same tool to be able to compare our data. We clearly show that lymph nodes present variation in number of variant and infected productive cells within the same lymph node, but also between different sites. Apparently, 5-micron section might not be completely representing a full lymph node, so there will be more data to come, but that's something to keep in mind. And what do we think also here in our lab is the microenvironment of infected cells or cellular reservoir could teach us a lot about the reservoir establishment, but also the persistence. And so few micron sections have limitation for that. And so right now we are working on a clear tissue assay to be able to assess the full microenvironment of those infected cells. So maybe next talk we'll have some data on that. I just want to spend the time to thank the people of the tissue analysis core. We are not an army, we are just the 4 of us. So this is Simona, Catherine, and David, and they are doing all the work that you just saw. So I will take any questions.